Few neurosurgeons have as much experience with treating glioblastoma as Dr. Mitch Berger. The CNS invites Dr. Berger to share some of his experience through his lecture, GBM, Adapting with Precision and Achieving a Change in the Landscape of Treatment Understanding of the Disease.
Thanks. Good evening, everybody. And I want to thank the Scientific Program Committee for asking me to give this talk. I don't have any disclosures other than the fact that Russ Lonser is a great friend of mine, and I wanted to congratulate him on a great year as president of the CNS. So I think it's fair to say that we haven't made a lot of great progress in this disease in the past several decades. In fact, if you look at the survival curves before Temodar and at the time of the first report in the New England Journal in 2005, which is about 25 years, the average increase in survival was really only about 4 months. And if you look at the tail, you say to yourself, well, what's the long-term survival of this disease? The tail of that curve is only 10%, so there's no question about it. We just really have done a terrible job in fixing this problem. It is a huge enemy of ours in neurosurgery, and we got to have better treatments. So enter the New World Order. And the New World Order is really the genotypic discovery and diversity and how this knowledge is going to be used to attack this disease. And my talk's going to cover a number of these areas and end with a brand new concept about how to go after glioblastoma. Let's start with the WHO. So this year, the WHO came out with its new classification. I was lucky enough to be the only neurosurgeon asked to this neuropathology fest, if you will, in Europe, and we developed a new classification scheme. And I think what's different about this is that the phenotype, or the histology, has now sort of emerged into giving way to the molecular signature of this disease. So it's a phenotypic, genotypic description. So for example, we can't use the term low-grade glioma anymore. There are really three different entities based on the status of IDH and the 1p19q situation or status. GBM, of course, is known in the primary setting as the wild-type IDH genotype. And of course, GBM has a lot of other somatic mutations and amplifications and deletions that really define this disease further. Now, about a year ago, our group at UCSF, along with our colleagues at the Mayo Clinic, put out what I think was a really critical paper. It was published in the New England Journal, and the goal of this paper was to say, look, can we boil this disease down to just a few markers? So we took the IDH and we took 1p19q and we added another new observation, probably the most frequent mutation in all of cancer, called the TERT promoter mutation, which affects telomerase function. And we put those three markers together and created 5 subtypes. We did this in San Francisco. The Mayo Clinic did it separately and with their patient population. And you can see here on the top 3 columns, these are the low-grade gliomas, and over on the right side, we see the IDH wild-type GBM with or without the TERT promoter mutation. And so why did we do this? We did it because we have to use a better strategy to predict outcome. In this new world order, you can't use histology anymore. You've got to go with the genotypic description of this disease. And sure enough, those patients who have that wild-type TERT promoter mutation, they're the ones that do the worst. And in fact, when you look at all of this together, you come to realize, as you look at these hazard ratios, especially in the bottom right-hand corner, that it's really the molecular genotype that defines this disease. This next generation, all of you, as you're coming up in the field, you're going to forget about histology. You're going to have to describe this disease based upon the genetic and genomic alterations that are present, because it's more predictive of outcome. You know, what's amazing about this disease, if you stop and think about it, is that if you look at where GBM is in the mutational landscape, the spectrum, it has fewer mutations in, say, lung cancer or colorectal cancer. So by definition, you would say to yourself, well, we should be able to fix this problem, right? It's not as complicated. But the reality is, it is extraordinarily complicated for a couple of reasons. First of all, we don't have any lack of new targets for this disease. This whole new world order has created a whole bunch of new therapeutic targets that are going to go beyond the era of chemotherapy. And we've got a lot of targeted therapeutics made by hundreds of pharmaceutical companies that are now engaged in the clinical trial realm. But here's the problem. This is one of my patients. I operated on this patient in that sort of greenish circle to the left. That was the primary tumor. The patient relapsed. I took him back to the operating room, reoperated. The patient relapsed again. I went after it again. 3 different operations, 3 different time points. But here's the problem. It's a moving target. Over on the left and on the right and on the top, in those 3 specimens in 3 different time periods, they only shared 32 genetic alterations, whereas there was a 9-fold increase in genomic alterations in each of these specimens that they didn't share. So how in the world are we going to attack this problem if it doesn't sit still? So enter the concept of precision therapy. We heard this from our president, who said it's time for precision medicine. So we're going to develop precision clinical trials, and it's based on the understanding that we can define individual heterogeneity in a regional and spatial fashion in these tumors. By getting samples from all over, we can now look at the targets that are out there. We can define which targeted therapies cross the blood-brain barrier and act on these actionable targets. But the problem is the clinical trial design that we have currently across the board doesn't work. And the other problem is we can't get one industry partner to talk to another one. Because if their drug fails in the face of another one, then everything could go down the tubes with their drug development. So it's a huge problem. So what did we do? We formed a consortium. We said, okay, let's get off the mark. Let's start with precision medicine. This was the early phase clinical trial consortium for precision neuro-oncology. We started it in San Francisco. We had a bunch of groups, some of whom the investigators are in this audience today. They joined efforts. And what did we do? What we did was we looked at this disease in terms of its heterogeneity. In other words, when I went to the operating room, I didn't take one specimen, I took 4, 5, 6. We looked at the heterogeneity. We looked at what targets were present. We looked at what drugs industry had for us, which drugs got across the blood-brain barrier, had favorable pharmacokinetics. We interrogated all of the molecular pathways. We put all this together and said, look, let's just take this disease and hit the heck out of it. Let's treat it with 1 drug, 2 drugs, 3 drugs, 4 drugs. Let's do this in a rational fashion. So what happened was a patient would come in, we'd take him to the operating room, we'd take out a piece of tumor from the enhancing area, we'd take it from the brain adjacent to the enhancement. We would do whole exome sequencing and RNA sequencing through TGen and Phoenix. We put all that together into a roadmap of potential therapy based upon the alterations present in that one patient's tumor, and then find out which drugs had the optimal pharmacokinetics. And this is what the map looked like for a given patient. You see the genomic alterations, and you see the drugs that are out there available to be treated. What was amazing about this study, and this is what we were told we couldn't do, is we couldn't go to the operating room and then within 30 days get all this information. Well, we proved these pundits wrong. We showed that we could do that. Well, we could take this tissue, we could sequence it, we could go back and have a genomic tumor board, and we did it in 94% of the population. And it's interesting that when we went to the patients and said, hey, here's a personalized treatment for you, do you want it? 50% of the patients bought in and said, yeah, I want it because I know nothing else is working. And the recommendations often involve these repurposed drugs, these drugs that had no role in neuro-oncology, but in the New World Order, they hit a target. And the average number of therapies per patient was a little bit under 4. And this is what it looks like. So 16 patients from top to bottom, across the board, the genetic alterations, not one patient shared the same genetic alterations with the other. All 16 were different. And sure, they clustered where you thought they would cluster around EGFR, P10, and the cell cycle genes. But the reality is we could take that information for a given patient, we could recommend treatments based upon the targets, and then we selected those targets with our genomic tumor board, and we treated those patients. Here's one example of how well this is working. This is a patient who failed standard therapy. They came back, had a relapse, they had an operation. There were 3 targets, 2 of which were pretty significant targets. Our regimen was designed with 3 agents that we never have heard about before. We haven't used other than the carboplatin, which had a specific target to hit. We treated the patient, and sure enough, that patient is still alive on targeted therapy 16 months later. So that's a very unusual and yet an extremely successful example of how this works. So where do we go from here? Because again, it's not working. So enter GBM Agile, and Agile stands for Adaptive Global Innovative Learning Environment. And what happened was a bunch of us got together, some of whom are in this room, and certainly myself as well, with the basic scientists, with the clinical oncologists, with the people who understood pharmacokinetics. We had think tanks, we had workshops, and we all sat in a room and we said, look, we know this is not working, right? And why isn't it working? Because if you think about it, we only have one predictive biomarker in all of glioblastoma that predicts response to therapy. That's the MGMT promoter methylation. There's just one. None of the targeted therapeutics have worked because we haven't looked at the individual profile of those patients. It was a shotgun approach. We have a lot of prognostic biomarkers, but we don't have the therapies yet, like the TERT promoter mutation. And the clinical trial designs that we're using just don't work. They're not adaptive, and they take too long. And we're getting real-time information as we move forward. This is a long laundry list of things, but just put up with this for a minute as I explain it. The goal of this Agile trial is to develop this prospective randomized phase 2 study for both newly diagnosed and relapsed GBM. And what it does is it randomizes against the standard of care. So we know what that is, right? It's radiation and Temodar, or CCNU relapse, but it's an 80 to 20 randomization. So you could have a whole bunch of experimental arms versus the standard of care. You're dealing with only one type of lesion. That's the IDH wild-type tumor. You stratify the disease based upon the promoter methylation, and there's a biomarker decision based upon the tissue. So in order to get in the trial, you got to have tissue from that patient, and the biomarker, which is the target, has to be linked to a targeted therapeutic. You can use 1 or 2 drugs if you want, but here's the beauty. It's adaptive. And what I mean by that is you kick out a drug if the drug isn't working. You keep a drug if it's working. You kick it out if it fails. If it's working, you keep it in, and you never close the trial. So that when a new agent comes along, you can add it to the trial, and it's all under this master IND protocol, which allows us to never close the trial, which is the complete antithesis of what we're doing today. So why use an adaptive trial? Well, we use an adaptive trial because the strategy is you want to learn from each patient. So the trial accumulates data obtained while the trial is ongoing, and the data is used to modify the course of the trial. You know, breast cancer now has been attacked with this adaptive trial method. It was the I-SPY 2 trial, which has been very, very successful. We think we can do this for glioblastoma. And he uses this very interesting, innovative statistical approach that allows you to build as you go and make the trial more accurate because it accumulates ongoing longitudinal information. You can start with many, many arms, but you don't need to use all those arms equally. So if something's working, keep it in. If it's not, throw it out. And this enables us to determine that not only the drug is safe, but it's effective, and it shows how it should be delivered and to whom should get the drug. Listen, that's the essence of precision medicine. This is what we have to be doing. So how's this going to happen? It's going to be an international trial. We've got Australia, the Chinese, the European Union. This is going to be a big trial to get sufficient numbers of patients to actually test the efficacy of these agents and combinations. The adaptive design will allow us to learn from each patient. So you enter a patient, you learn how they're doing, and then it informs you about the next patient that enters the trial, and the effective drugs graduate with their peculiar signature. They have to have a signature, meaning the molecular piece, which increases the probability that they're going to succeed in a phase 3 trial. And remember, the trial never stops. It keeps going under this master protocol. So one of the conundrums is going to be, how are we going to pick the agents? We're going to pick the agents because we're going to have a standard level of evidence based upon previous safety data, the prior history that it was in some trial, that it has a target that it can hit, that it has favorable pharmacokinetic data, and that there has been some efficacy in prior studies. We're going to have an agent selection committee that's going to determine whether or not the drug is approved or disapproved. So here's essentially my last slide, and let me just show you the workflow of this thing. And then you'll see the logic, and you'll say, why haven't we been doing this before? So you see a patient in the clinic, looks like they have a glioblastoma. You take them to the operating room, you operate on them, but you sequence that tumor. So you find out what their molecular subtype is, and then you randomize them to either go on an experimental arm or a control in the 80-20 fashion. The outcome data is collected as you go along, and it's updated through this longitudinal model that allows the statistics to tell you when the experimental arm is better than the control. And there are decision rules in all of these arms. So if a drug's not working, you pull it out, you go back to industry and say, here's your drug, it's not working. But if it's working, you keep it in the trial. And if you accrue 150 patients, if that's your trial accrual, then you stop. And then what you can do is you can graduate that drug that's succeeding, and you can make it into a phase 2 or a stage 2 registration trial so that it can get FDA approval to use. But the beauty here is that as new things come along, as people in this audience come up with new discoveries from their labs, we can add new experimental arms. We can continue to calculate the probability that the experimental arm is better than the control and determine the adaptive randomization probability within each molecular subtype. It's a great design. It worked for breast cancer. It's got to work for us. So the bottom line is we are in the New World Order. This is a very, very formidable disease. We all get it. But we have to capitalize on the evolving genomic landscape of this tumor and what we're learning from it, because in essence we have to build a precision-based individualized therapy that's going to treat this disease like we've never treated it before. So stay tuned. I think this is going to do it. I really do. Thank you.